Bradley Mesmer. I'm the founder and CEO of Abreos Biosciences. We're a San Diego-based company, which means I have a little bit of jet lag, but hopefully I'll get through this. We were a 5.5-year-old company based on technology that was originally developed in my lab at the University of California in San Diego, where up until recently I was faculty in the Cancer Center. And we're raising a Series A of $10 million, or 10 million euros, to develop this integrated therapeutics story that I'm going to tell you about. But before I tell you what we do, let me tell you why we do it. So this is Mary. She's typical of a patient battling MS. And 10 or 20 years ago, a diagnosis of MS typically comes in early in life, would mean that a patient like Mary is looking forward to declining function that could ultimately end in a wheelchair or worse. But today, there's a number of disease-modifying therapeutics, most of them biologic drugs, that can really halt the progression of this disease so that patients like Mary can live very, very full lives. Now, these drugs, they really work, but they come with costs. First of all is the actual costs. Obviously, we pay more in the US, like we do for everything in healthcare, but even in Europe, these are very expensive drugs. And at least in the US, patients like Mary often have relatively poor insurance, and the average MS patient in the US bears about a third of these costs personally. But more worrying than the actual money costs is the potential for very serious side effects. All of these drugs modify the immune system and as a result make those patients at high risk of infection. And in the case of Tysabri, PML is a brain infection that kills 1 in 4 people that develop it. So while these drugs really work, the patients on them have to be very concerned. And we now know from a variety of clinical literature that risk is related to the way that these drugs are given. Like many biologic drugs, they're given in a one-dose-fits-all strategy. And that turns out to really be a problem because what we know is that there's enormous variation in the pharmacokinetics of biologic drugs. So if you look at how much drug is in the blood of a treated patient, from patient to patient, that can vary by about 100-fold. In fact, that's true for all of these drugs. And the traditional paradigm has essentially been to massively overtreat everybody to make sure that you cover the efficacious range for all patients. And so what our company is focused on is personalizing the dosing of biologic drugs, which will have multiple advantages, but most significantly, lowering the side effects as well as lowering costs by not using more drug than needed. And so how do we do that? Well, that's our core technology. This is what we call the Veritope platform. Veritopes are peptides that we select from phage display peptide libraries, and they bind specifically to a given biologic drug. So we make a peptide that binds to the molecule and can then be used as a sensor in a variety of immunoassay platforms. We routinely develop laboratory-based assays, but what's particularly exciting about using these peptides is that we've been able to transfer them to point-of-care platforms, most notably lateral flow immunoassay. So we're able to create with these peptides tests that are essentially like glucose monitoring, but in the form of something that would look like a digital pregnancy test, where a drop of blood applied to a paper strip can give a quantitative readout of the drug level in that drop of blood in a matter of 15 minutes for a device that costs under 10 euros to produce at scale. So as I've mentioned, this technology came out of my lab at UCSD. It's covered by an issued US patent that broadly covers the use of mimicking peptides to measure biologic drugs, and then we filed a number of patents around specific variations on the assay. We've been supported extensively by the NIH. We've had over $5 million in NIH grant funding over the life of the company. And we're actually poised to launch our first commercial diagnostic assay for the drug Tysabri in MS as a laboratory-developed test, which in the US does not require FDA approval. With that said, we've actually just signed a research collaboration with FDA, who approached us to develop assays for some other drugs that they were interested in looking at lowering the doses of. So now I'm going to tell you a little side story that will lead to hopefully the punchline. So as I said, we've been active in the MS space for a while, and it's interesting that the old cancer drug Rituximab has actually been known to be efficacious in MS for about a decade. However, Roche chose not to get it approved and instead developed Ocrelizumab, which works essentially the same, but has different economics. Now, both of these drugs work by killing B cells, and there's extensive literature showing that the dosing is excessive. So what's more, Rituximab is now off patent, and there's a number of biosimilars being developed in the US and Europe. But again, it was never approved in MS, so by definition, a biosimilar cannot be used for MS. So our concept is to take our dose monitoring and couple it with rituximab to create what we're calling an integrated therapeutic, where the personalized dosing will be part of the clinical trials and therefore part of the label. So essentially creating a novel therapeutic from a tried and true drug component, but personalized through our technology. And so the way that this will work in practice is as follows. All the patients will be started on a standard dose, and then essentially we will do a careful dose de-escalation based on what's actually happening in that patient as monitored by our technology to gently bring them down to a stable maintenance dose for that patient. So this scheme, we believe, creates some novel approaches to value in this therapeutic space. It's not just the drug, it's the integrated therapeutic that provides benefit, and it does that on the payer side because we can come in at a cost that's well below the existing therapies, but it also then drives adoption to the patients and physicians because they want personalized therapies, they want to lower their risk of side effects, and they want to spend less time in an infusion chair getting their medication, which they will since we don't have to give them as much. Now, on the business side, this is a big opportunity. The MS market is large and growing, and the biologics are capturing an increasingly large fraction of that market. So we project that even though this is a sort of repurposed therapeutic, it's still a billion-dollar opportunity. And this is based mostly even just on the US. If you add Europe, it's even larger. So the team that's bringing this to fruition is led by myself. I'm joined by Michael Williams, who is an ex-pharma guy. He was head of global marketing at Takeda most recently. He's led a number of biologic drug launches. Helen Jenkins is an experienced operator who has been part of a couple very successful biotechs with big exits. We're joined by our VP of Finance, who is an old serial entrepreneur, and we have some younger entrepreneurs and scientists on the management team. Our advisors are really important. We have key opinion leaders in the MS space. Dr. John Foley is actually one of the first investors in Abreos. When he saw what we were doing, he said, "I've been looking for this for years and I want in." Mark Backer was the 7th employee of Genentech and actually made the first batches of rituximab. He now runs a CMC consultancy in the Bay Area and has been a longtime advisor to the company. And we have a research subsidiary based in Marseille that's incubated in the IPC Cancer Center there, and Dr. Saunier leads our efforts there. Just as a small aside, we're also looking to spin out the pure diagnostics business into a European entity, and we're talking with people in France and Portugal. So if anyone's interested, please grab me. Now on the IP side, as I said, we have a patent covering our way of measuring things, but more importantly, because this will have to go through a new drug pathway, we will get the data exclusivity that's given to any BLA filed drug. So the barriers to entry here are as much a kind of a first mover advantage as well as the clinical data, not so much just the raw formulation of matter. With that said, there's also extensive opportunities to continue to innovate on the diagnostic and the integration side, providing for evergreening of IP. Now, this isn't just a one-off opportunity, although we do think this is the right place to start. There's a number of other indications in which rituximab has been used successfully off-label that would benefit from this dosing, as well as the indications where it is on-label. And then looking forward, a very large fraction of the existing biologic market is losing patent exclusivity or has lost it already, creating multiple opportunities for us to apply this paradigm where personalized dosing can take a drug you already know it works and just make it work better. So as I said, we're raising a $10 million Series A that will do 3 critical things. It'll allow us to develop our rituximab or in-license one. We are in discussions with a number of biosimilar manufacturers. It'll allow us to bring our assays up to snuff for regulatory approval because the assay will be joined, the diagnostic component will be joined at the hip to the therapeutic. They have to go through an approval process hand in hand. And most importantly, we'll do a pilot study using off-the-shelf rituximab, which we've already designed with Dr. Foley, That will validate that these lower doses have the primary biological effect that we believe they will, and allow us to assess the variation that we already know exists, but we'll see it in our own clinical data. Then that will lead to a phase 1/2 combined study that will be funded by a Series B. Obviously, going through a full drug development path is an expensive proposition, but we think after the phase 2 data, we will have opportunities for exit or partnering. And we do actually have a bridge finance note open right now to address some short-term capital needs. So I'm happy to discuss that in more detail. So to summarize, we think that this is a really powerful approach to create best-in-class therapeutics without the clinical risk associated with novel molecules. We're taking drugs we already know they work. There's 10 years of data on Rituximab. It's the drug of choice in Sweden, among other European countries, because of the lower cost. So we know it's going to work. We know we can make it better, and then we can replicate this model across a variety of indications. And so I'll just end by saying that this is not really a new idea. 500 years ago, the connection between dose and effect was pointed out by Paracelsus, so it's about time it came to the biological drug market. Thank you.